Hello, everyone. Welcome to the webinar MP101 for Asia Pacific and EMEA. My name is Bhaskar Barnwal, and I am the field application scientist based out of Singapore. And here is my email ID, which is given there at the down. So today we will learn about mass photometry. Some of you guys might be aware about it. Uh, so let's go and see the agenda for the talk today. We'll be talking about Refeyn in the beginning, just a little bit introduction about Refeyn and the principle behind mass photometry, which will be followed up with the key applications and question and answers. So in the Q&A session, it's possible that I might not be able to answer all of your questions because of shortage of time. So you are basically requested to either reach out to me or you can send your emails to support@refeyn.com. Okay, so let's get started. Refeyn was spun out from University of Oxford in the year 2018, and in just 7 years, we have installed more than 500 instruments in 30 countries. We have a portfolio of around 58 patents and more than 1,000 publications featuring mass photometry. It's a major milestone for us, and we are proud to say that we are present in around 18 out of top 20 biopharma companies in the world. Biophysical characterization aims to answer questions about the fundamental nature of biomolecular behaviors, and these questions broadly encapsulate three areas. Sample characterization, where we are considering the sample as a whole; analysis of complexes, where the focus is on deriving structural information about a particular species within that sample; and interaction studies, where the area of interest is how a particular species interact with others within the sample. So today I'm here to introduce you to an innovative addition to our biophysical analysis toolbox, which will help address these key questions. Mass photometry. So what is mass photometry? Mass photometry delivers rapid, accurate mass measurement of label-free single molecules in solution to their native state. And the ability to measure these mass of single molecules in a bulk sample means that mass photometry can provide access to subpopulation to determine complex stoichiometry and oligomeric states. It also means that it is possible to study the interactions and even complex multi-step processes which are accessible by mass photometry. Furthermore, the single molecule counting over a wide mass range enables the detection of low abundance species and the characterization and monitoring of sample heterogeneity. We at Refeyn believe that this technology can help accelerate science, and by making characterization more straightforward and making it possible to analyze complexes, assembly, and interactions in new ways. So there are two mass photometry technologies. The first one is the core technology, where mass photometry measures the mass of smaller particles by measuring the contrast. And the second one is where it's called macro mass photometry, and it builds upon our core technology, which measures the contrast of the particles. Along with that, we measure the size of the particles as well. These are basically for large particles such as adenoviruses and lentiviruses, apart from virus-like particles. So the machine which is based upon macro mass photometry is CaRiTro-MP, which ranges from 40 to 150 nanometer in diameter. And today we will not be talking about this one. We'll be majorly focusing on the mass photometry today. And the 2MP machine is the one which is based upon mass photometry technology as the SEMEX-MP. The difference between the two is that the 2MP has a wide range from 30 kilodalton to 5 megadalton, which can cover the applications having protein complexes, nucleic acids like RNA and DNA and plasmids, macromolecular assemblies, and nanostructures. However, SAMXMP is mainly focused or is designed for analysis of AAVs specifically, and we can have a look at the partial and overfilled AAVs as well using SAMXMP, apart from getting the AAV titer from there. We also have the GMP compliant softwares for the SAMXMP, so you can analyze it in good manufacturing practices. So what you see on the screen is the molecules which are landing, is a single molecules which are landing onto the glass cover slip in PBS buffer. In this particular case, it is thyroglobulin molecule, which is of 660 kilodalton molecular weight. The landing events are actually recorded by our ISCAT technology, also known as interferometric scattering microscope technology, which measures the contrast values generated due to landing of these molecules because of the interference between the reflected and the scattered light. To know the biophysics in detail, or to know the core principle in detail, we will go to the next slide, where we will try to understand about what is happening during the measurement. So in this particular picture, what you see is a glass cover slip, and this glass cover slip is illuminated by a laser light from underneath. You can take your samples, which range from DNA, RNA to proteins, and even AAVs on top of it. Some of the light goes through it, while some of the light is actually reflected back due to this glass buffer interface. And some of the light is actually scattered due to these landing molecules. The scattered light and the reflected light then goes back through the objective lens, where it interferes with each other constructively, leading to these contrast signals, which we have seen in the previous slide. Now, there is an interesting phenomena here. The contrast signals are linearly related to the molecular weight of these particles. What it means is that if the particles are heavier, then the center of these signals will be darker. And if the molecular weight of these particles are lower, then the center of these signals are dimmer. So we take advantage of calibrant whose molecular weight is known, and we draw a nice standard curve between the contrast and the mass. And at the end of the day, what we get is a histogram between counts and the mass, counts being on the y-axis and mass on the x-axis. What you get from here is the molecular weight and the relative abundance of all the species present in the sample. So that's the core principle behind it. And with this, let's see what does a mass photometry measurement look like. So this is the video of mass photometry measurement. The glass slide is placed on top of the objective lens, and the sample is then added to the buffer drop, and then it is mixed gently. Once the lid is closed, then you will see the molecules landing onto the glass cover slip, which will be like this. So mass photometry is versatile because it measures mass, and it's a universal property that can provide useful information on many different type of samples. So it can be used in many different ways. So let's get started with sample characterization here. Mass photometry was used to check the monodispersity of the HTT-HAP40 complex, HTT being a gene which is altered in Huntington's disease. And this study was done prior to further biophysical analysis, and it confirms, the mass photometry study of this complex confirms that it forms a very stable non-covalent complex. This data was published in the year 2021. And if you have any assessment for the proteins, for the purity or the quality of the protein, you can always use mass photometry for that thing. This particular work was published in 2021, and here they assess condensin 1 complex in different conditions like temperature and sodium chloride concentration. Just two are shown here in this picture. The first one has 50 millimolar sodium chloride, and the second one has 150 millimolar sodium chloride in the buffer. As you can see, condensin complexes remain intact in these two conditions. So if you are studying your complexes in different buffer conditions, you can always do that using mass photometry. APCC, which is anaphase promoting complex cyclosome, is made up of 19 proteins, and its production involves a three-step purification process, which is streptactin chromatography, ion exchange, followed by size-exclusion chromatography. The mass photometry data which we obtain aligns very well with the NSEM data, as you can see here in the mole fraction. It aligns very well, especially these fractions. These three fractions have higher proportion of the complex in the sample. So mass photometry gives quick and clear information on complex assembly using very little sample, while in NSEM you might need more sample and it might be more expensive to do the experiment. So if you are studying aggregation or fragmentation, you can always do that by mass photometry and as a part of antibody stability. In this particular example, our industrial partner gave us 14 different antibodies, and it was subjected to 3 different stress conditions along with a control, and it was run on mass photometry in triplicates. So in total, 168 measurements were done. And as you can see, in the case of candidate 1, in the stress 2 situation, the amount of fragments or the proportion of fragments you can see here is higher in comparison to the control. Also, for the candidate 2, you can see in the stress 1 and stress 3 conditions, the dimer and trimer proportion is higher in comparison to the control. So if you are studying the antibody stability and interested in aggregation or fragmentation of your antibodies, you can use mass photometry to quickly analyze your samples. So this was trastuzumab originator molecule, and it was studied by RIC Biologics, and 4 different clones of trastuzumab originator were evaluated. This one, the left picture shows the data from mass photometry, and the picture on the right shows the SEC-UV data. As you can see here, the clone number 3 and clone number 10 has higher proportion of aggregates, which we also see in the SEC-UV data. What we see is the clone number 8 and clone number 9 is having the aggregation which is very similar to originator molecule. That means more molecules are in monomer form, and that's what we see in the mass photometry data as well as here. But we can get similar kind of data with SEC, just that we have to spend less amount of sample. In this case, we used 30 nanogram of antibody sample. In SEC-UV, it was 10 micrograms. So it is 310 times lesser sample. And we did it 24 times faster because each analysis can take around a few minutes for mass photometry. However, for SEC-UV, it might take longer time. When a candidate moves to later stages of development in preparation for regulatory submissions, a column-free method for antibody aggregation needs to be run, and AUC is normally used for this. Control and heat stress samples were compared here by mass photometry and AUC. What you can see here is mass photometry is able to resolve 4 aggregates here. However, for AUC, you can only resolve the dimer to the baseline resolution. What you also see here is that the runtime and the sample use is very little for mass photometry. However, for AUC, you have to spare more sample and more time. So for the resolvable peaks, what you can see here is that for the dimer and trimer proportion, the MP and AUC gives very similar data. The data aligns very well for the control and the heat stress sample. So we can say that we can get something where you can use it as an orthogonal technique to AUC and get the data much faster than AUC. Since we measure the mass of the particles and not the size, we can easily differentiate between empty and full AV particles. Mass photometry can accurately quantify full proportions within minutes with very little sample. Remember that SAMX-MP can provide you more data by providing insights about the partial AAV and overfilled AAVs, apart from giving you the AAV titer estimates. And we also have the GMP compliant softwares for SAMX-MP, which is not there for 2MP. Well, we can also analyze RNA apart from proteins. We can also analyze the nucleic acids, as mentioned before, like DNA and RNA. Here I have taken an example of RNA. Usually the capillary electrophoresis or HPLC assess the mRNA purity in reducing and denaturing conditions versus mass photometry where we use native conditions. So you can see the manufacturer's certificate of analysis here, and the monomer percentage of RNA is around 80%, as you can see here. We can run the mass photometry either in the denaturing conditions by denaturing our sample with urea, or in non-denaturing condition, which is the native state. So we denatured intentionally to see if we can get similar proportion as we get in the certificate of analysis. And as you can see that monomer proportion is around 82%, which is quite comparable to what we get from the capillary electrophoresis. When we ran it in the non-denaturing conditions, which is in the native conditions, what we also get is the monomer, dimer, and trimer proportions of the RNA, which shows that the RNA exists as dimer and trimer as well, which we were not seeing in the capillary electrophoresis, which is generally run in reducing and denaturing conditions. Similarly, the next data is provided by Genentech, and they analyze it in the native buffer. What they could see is for 3 different RNAs, which is EGFP, FLUC, and beta-gal, they could see the dimer and trimer proportions if it was there in the sample. And they also analyzed the stability of the RNA apart from the purity. And as you can see here, for the mRNA sample, they heated it at 37 degrees centigrade for 5 days and had a control sample run just before analyzing the heat stress sample. You can see the control sample having both monomer and the dimer proportions. When the heat stress sample was run on the mass photometry, you can see a drop in the proportion of dimer almost disappeared there, and a new pre-peak region appearing just before the monomer. So if you are analyzing your RNA for your stability and purity for whatever reason, it could be for therapeutics, it can be for vaccines, you can always do so using the mass photometry very quickly with very little sample. Okay, so let's move on to analysis of complexes, where we will try to understand where mass photometry can be quite useful. And this is the example of 70S ribosomal complex. And it was analyzed in presence of magnesium and absence of magnesium in the buffer. As you see here, when the magnesium is not included in the buffer, you do not see any complex at 70S, and you only see the 30S and 50S complexes. But when you add magnesium into the buffer, you, apart from seeing the 30S and 50S in the sample, you also see a 70S complex at 2100 kilodalton. So, well, you can play around with the buffer and you can try to understand your complexes. You can try to see which buffer is suitable for the complex assembly, and you can optimize the buffers as well. This particular example is where Rubel 1,2 interacts with RPAP3 to form R2-TP chaperone complex, and it was used to track the assembly of complexes. The RPAP3 was titrated in different concentrations. However, Rubel 1,2 was used at a constant concentration of 0.2 micromolar. When there is no RPAP3 in the sample, you can see the monomer and the dimer of the Rubel 1,2 complex. When the concentration of RPAP3 was increased to 0.4 micromolar, what you can see here is that the dimer of the Rubel 1,2 disappears, and you can see all the 3 stoichiometric ratios, like 1:1, 1:2, and 1:3 complexes appearing here in the sample. And when you increase the concentration of RPAP3 by 4 times, what you see is the dimer is still not seen, And at the same time, the 1:1 stoichiometric complex has gone down in proportion. And you see the increase in 1:2 and 1:3 complexes here. So if you have any complexes and you want to do any titration studies, you are always welcome to do that using mass photometry to understand about your complexes. Okay, so membrane proteins are a pain point for a lot of researchers, and we know that membrane proteins are traditionally very challenging to measure. In this particular example, BO3 oxidase protein has been used, and it was stabilized with LMNG detergent. And just before doing the analysis on mass photometry, it was diluted in detergent-free buffer, and then it was analyzed on mass photometry. As you can see here, we can see the BO3 oxidase monomer at 290 kilodalton and the dimer at 506 kilodalton. Now, earlier it was debated, the dimer existence was debated, but mass photometry has kind of confirmed that the dimer proportion exists, and it is like 20% of the complex is present in the dimer form. And the data was published in 2021. Please note that the monomer size is 144 kilodalton, but the additional molecular weight which you see here is because of the LMNG detergent binding to the complex, binding to the monomer. All right, so you can study the detergent membrane proteins with the help of detergents, but if you have something like where you can embed your proteins into nanodisc, like in this case, it was embedded in small p, you can also use amphiphol. So you can use that and you can study those complexes in nanodisc. So in this particular example, aquaporin 4, which is a water channel, was embedded in small p, and then it was purified by size exclusion chromatography. After that, different fractions was analyzed by mass photometry, and as you can see, the fraction number 3 shows higher proportion of the small p embedded AQP4 tetramers. And it was followed up by antibody binding assay to show that the AQP4, which is embedded in small p, has been natively folded. So it was allowed to bind or incubated with the antibody against the anti-AQP4 antibody. And as you can see here, the AQP4 binds to the antibody, and you can see another peak for that one. So due to its rapid testing and compatibility with the membrane mimetics, mass photometry can expedite your membrane protein purification prior to further studies for structure and function. Mass photometry can also detect polymer formation, as in this case, it is arginine decarboxylase, and it was evaluated at 4 different pH. You can see that at highly alkaline pH or at the acidic pH, the complexes do not form, they disassemble. However, at pH 5 and 6.5, you can see the complexes which are there at around 900 kilodalton. This was also confirmed by cryo-EM. As you can see, the decamer from the cryo-EM. So the authors of this paper concluded that enzyme polymerizes in a pH-dependent manner, and that's what we are seeing here, that at pH 6.5 and pH 5, it forms a complex. However, at a lower or higher pH, it does not. We can also do the low affinity interaction study using mass fluidics SC. In this particular case, mass fluidics SC was employed because the affinity of the interaction was very low. And we have used it to compare it with the manual dilution method. As you can see here, when the manual dilution method was compared with the rapid dilution using mass fluidics SC, you can see the complexes in the mass fluidics SC in the rapid dilution in 1:1 and in 1:2 ratio. You also see the dimer of the antigen. However, in the manual dilution, you do not see those complexes or the dimer of the antigen. So the rapid dilution by mass fluidics SC can be used for understanding your complexes or interactions when they are low affinity interactions. You can also run at higher concentration if you are studying your antibodies and you want to have a look at the aggregation of your antibodies at higher concentration, you can always use mass spectrometric SC for that. Moving on, we will be looking at some examples from interactional studies and try to understand how it can help in complex stoichiometry and understanding the binding affinity. So here, what you see is a Herceptin molecule and HER2 antigen, which has been run alone in the mass photometry. So this is the HER2 antigen, and this is the Herceptin antibody. When they were mixed in 1:1 molar ratio, what you see here is a complex of 1:1 stoichiometry with very little of 1:2 if it is there. But when it is mixed in 1:2 ratio, molar ratio, then what you see here is that the stoichiometry shifts towards the 1:2, and you see a major peak of 1:2 stoichiometry with very little of 1:1 stoichiometry in the picture. You also see a lot of excess of antigen here in this one, as we have added more of antigen in this particular reaction. So not only the stoichiometry, we can also give the binding affinity estimate for the interactions. In this particular case, our industrial partner provided us with 5 different antibodies with low, medium, and high affinity towards EGFR. And the binding affinity was calculated for both the different stoichiometries using mass photometry, and it was compared with the BLI. So what we found out is that mass photometry affinity measurements agree with the BLI measurements. And we also find out about the information of the stoichiometries. As you can see here, we can find out the proportion of 1:1 and 1:2 of the complex. However, we are not able to get this kind of data from BLI. So you can always use mass photometry to determine the KD estimate, and you can also find out the stoichiometry. It can be used as an orthogonal technique to BLI. So this data was done in collaboration. We obtained this data in collaboration with Absolute Antibody. And here is a panel of bispecific antibodies which were tested for their ability to bind HER2. As you can see in these pictures, the stoichiometry of the antigen-antibody interactions has been determined within minutes without any method development steps. So you can see the 1:1, 1:2 stoichiometries in these 4 pictures, and in these 2, you can see 1:1 binding. So it's a pretty rapid method of getting the data without developing any new method for that. So as I mentioned before, this mass photometry is specific. It has a range from 30 kilodalton to 5 megadalton. So we cannot study the small molecules directly. But in this particular case, we are studying, actually AstraZeneca has studied this, and PROTAC is a small molecule, bifunctional molecule, which cannot be directly studied since it is around 1 kilodalton in size. But since it can be used to bind to the E3 ligase and the target protein, it can be studied with mass photometry. This is a titration study where MZ1 was used from 0 nanomolar to 1200 nanomolar. And as you can see here, when there's no MZ1 PROTAC in the sample, you can only see a single peak. Unfortunately, their molecular weight is very similar, and that's why you can only see one peak for target protein and the E3 ligase. But when we add the MZ1 to the sample, what you see here is a ternary complex. When we increase the concentration of MZ1 to 100 nanomolar, apart from the ternary complex, you can also see the quinary complex. And when the MZ1 was increased to a very high level of 1200 nanomolar, saturation of the binding sites happen at the target sites, and thus it inhibits the complex formation, and that's why you do not see any complex forming here. So if you have any small molecules which can bring upon any structural changes, and it is bigger than 30 kilodalton, you can always study that using mass photometry indirectly. Whether it is glue or a small molecule inhibitor or a PROTAC-like molecule. So to sum it up, mass photometry is a single molecule technique on the bulk sample, which gives us information on oligomeric states, complex stoichiometry, and beyond that, makes the study of complex multi-step processes and equilibrium measurements possible. The single molecule counting over a wide range means that information on purity, sample homogeneity, and the detection of low abundant species is readily determined. The fact that sample is in the native state, in solution with no need for immobilization, we can, so we do not have to immobilize our samples like we do it in SPR or BLI. It simplifies the sample preparation procedures, and it means that the data collected is an accurate measure of true native behavior of the biomolecules. So that's mass photometry and the summary about it. At last, I think this is my last slide. Uh, I would like to again sum it up with the family of mass photometers which we have here. The 2MP, which has a range from 30 kilodalton to 5 megadalton. We do have an auto system which can measure 24 samples in 90 minutes. SAMXMP has been specifically designed for AAVs, which has a GMP compliant software as well, and it ranges from 500 kilodalton to 6 megadaltons. The resolution in the range of 3 megadalton to 6 megadalton is has been optimized so that we can study AAVs here. And we also have the auto system, which can do 24 samples in 90 minutes. And the last one is the macro mass photometer, which is CaRITRO-MP, which can be used to study large viral vectors like adenovirus, antiviruses, and virus-like particles, which is within the range of 40 to 150 nanometer. It offers a semi-automated workflow. And that's all. I think we will move to the question and answer session now.